Hello and good evening, everyone. I'm Emanuele Di Angelantonio. I'm a professor of donor health and director of the BTRU in donor health and genomics here at the University of Cambridge. And it's a pleasure to host you here for our seminar at the Cambridge Science Festival. So thanks for coming, first of all. Thanks for giving up your time despite the glorious weather and the glorious evening in a while, and the ability to go at least to be out of our house. So it's a pleasure to provide this seminar together with Dr. Lois Kim and Nick Gleadall, and we hope we will develop an interesting topic about blood donation and blood donation research. So next slide, please. I'll maybe make a start by describing a bit what is NHSBT. That actually is the main structure that actually collect and deliver blood in England. And as you probably know, so the NHSBT is actually responsible for three main functions. One is really about the blood supply, the second is really about the organ donation transplantation, and the third one is really about the diagnostic antibiotic service. And here today we're really focusing about the blood supply. And you know, there's about 1.6, 1.7 million donation per year that the service is collecting. There are about 900,000 donors, just under 1 million donors donating each year. And there are many, many thousands of sessions that actually collect blood every day in England. And if you can go to the next slide, please. Yeah, and one of the main function actually is of the blood service really collecting blood. But while collecting blood, one of the main issue that we're actually having is iron loss. So every time somebody donate a pint of blood, and every time there's a pint of blood donated, there's about 200 milligrams of iron actually lost in the bag. So how do we make up that the iron loss, and what is actually the effect of doing that? So of course, every time there's there's loss in iron, there's an increase in your body to increase iron absorption, and this is really true mainly through the duodenum, and actually is quite limited per day. And so usually there's a depletion of iron stone that is not actually put it back, actually can lead to different potential side effects. The most important one, of course, is the ineffective blood cell, especially red cell production, and so that can actually lead to anemia. So many individuals actually have anemia, usually have as well iron deficiency that actually could lead to that anemia. But as well, there are potential other effects of iron loss and iron depletion, and those are what we're actually being very keen to study and to trying to understand what is actually the effect of losing the iron during the blood donation. So if you go to the next slide. So over the last few years, our unit, together with NHSBT and University of Cambridge, we actually set up a different study really with the aim to try to study both the medium and as well the long-term effect of blood donation, and how actually that iron loss can actually have an effect on immediate but as well long-term future health of each donors. So if you go to the next slide. And so one way to try to do that in some way is doing that in an indirect way. So one of the things that actually vary across Europe and across different blood service is the time between donation. So as you can see from this slide, there are many different countries that have many different times that allow donors to come back for donation. So in England, the one with the red arrow, you can actually see that it's about 16 weeks for women and 12 weeks for men. But this interval is quite varying across different services. So there are many countries actually allow donors to come back much more frequently, up to 8 weeks in many countries in Europe, but as well in US, and between, let's say, 8 actually to 10, 12 weeks in women as well in many countries across the world. And so here's really sort of one of the key questions that we've been trying to answer is how can you actually vary that blood donation interval, and really how can that impact on first the blood supply, but as well on the iron store of each individual donors. So if you go to the next slide. And so really sort of the question is really how can we identify the optimum donation interval between donation, really to try to minimize the iron deficiency on one side, maximize blood supply on the other side, and the others as well sort of maintain the well-being of the donors. And for this reason, we actually set up the interval study. So if you go to the next slide, interval study is a randomized trial that actually is aiming to assess different outcome in blood donors. The first one, the key primary outcome, is about the number of the pint of blood donated, so really sort of the amount of blood donated by each donor. The secondary outcome was really about the donor's well-being, so potential side effect of blood donation and iron deficiency, and as well as a set of different secondary outcomes related to potential iron loss, sort of cognitive function, physical activity, potential cardiometabolic disease, but as well what is the cost effectiveness of using this approach compared to using the standard approach that actually the blood service is using. So if you go to the next slide, please. So the study was is one of the largest trial have been conducted so far in England, and actually is the first time that the trial was done in blood donors to assess the donation interval. And this is recruited basically 50,000 donors across 25 centers across the country. We decided to divide people in 25,000 men and 25,000 women, and each of them were randomly allocated to 3 different groups. One being the standard group here in blue, and the other 2 sort of the shorter group. So for men was 12 weeks as a standard group, and then 10 and 8 weeks as a shorter. And for women was actually 16 weeks, and then 14 and 12 weeks as a shorter interval. And so the decision why to go for this shorter interval was really based on, as I showed you in the previous slide, on what is the practice in other countries. Actually, some of them allow 8 weeks for men and as well 12 weeks for women. And the way that we do that was sort of through randomization. So that means that every time a donor was coming in the donation service, we're actually randomly allocated to one of these groups. And then they were supposed to follow that interval for a duration of 2 years initially, and then extend it for another 2 years. So if you go to the next slide. So the trial was quite successfully in recruiting. We spread the recruitment across the country. You can see on the left-hand side the different area from which different center from which we recruit, and actually where's the majority of the donors have been coming from. This is quite a good reflection of what is the general blood donor population. So on the right-hand side, you actually have the distribution of the donor population across England, and with a focus on London. And you can see that actually, we were able to scatter across the actual recruitment across the entire in some way coverage of the NHSBT service. So if you go to the next one. So what was the main results of the study? So once we randomize people to the different interval, then we follow them up, as I say, initially for 2 years. And then we've been evaluating how much blood they've been able to provide over this time period. And so here you can see 2 main plots that represent the main results of the study in men on the left, women on the right, in which we actually compare the different time between donation and how much blood are being provided over the 2 years period. And so you can see that moving from a 12 weeks interval to a 10 weeks interval to an 8 weeks interval for men, there's actually an increase in blood donated over this period. And actually similar as well in women, which moving from 16 to 14 to 12 was possibly less steeper increase in blood donated, but actually again a sort of a significant increase in blood donated by women over that period with the shorter interval. In the blue line here as well, we are representing what was actually the amount of blood that were donated by donors in the 2 years before the trial. And this will give us an interesting comparison for 2 reasons. One is actually you can see that the people were randomly allocated to each group. Doesn't mean that there's no preferential people that were used to donate more that end up in one or the other group. And the second one is actually give us a baseline that even if those people were allowed to donate at 12 weeks, even just by being in the trial, we're actually able to donate more. And this was really due to the fact that these people were motivated to come, were actually invited in different text messages and called really to stick with their appointment. And the same picture for men and same picture for women. So both the fact of being in the trial, but as well in terms of being a different interval, provide a much increasing number of pint of blood donation during 2 years. So if you go to the next slide, that was the positive side of the coin. So yes, by decreasing the interval between donation, the blood service can actually collect more blood, and that's from an efficiency of blood collection can be very good. What is the potential side effect of that? Here's one of the main potential side effects that we recorded during the trial. So as we expected, during the main trial, if you at the end of the trial there was a decrease general decrease in hemoglobin level in every group of donors actually donating. That's what we expected. But actually, this decrease was greater in people donating more frequently than people donating less frequently. So you can see from the green line, at the end of the main trial, the green line is 12 men donating at 12 weeks. So actually, the hemoglobin level was on average higher than people actually donating every 8 weeks, always at the end of the trial. And this actually was the same when we prolonged the trial. So this is when we prolonged the trial for another 2 years to see the longer term effect of changing the interval. And you can see as well that this difference was maintained. And similar for women. So women donating less frequently have a higher hemoglobin level compared to women donating more frequently. And the same picture was at the end of the study. So if you go to the next line, as expected again, once you see that the decrease in hemoglobin level, there was a corresponding decrease in the iron store. And so you can see that there's a huge drop from the baseline to the end of the main trial, and then actually was stabilized as during the extension phase. So actually that with a large difference between the different groups of donors donating a different interval. And this is the same, roughly the same for both men and women. So if you go to the next slide. So that was in some way the negative side of increasing blood donation, increasing the frequency of blood donation. Was the other positive side, actually, that there was no major effect on that decrease in hemoglobin level and iron in terms of quality of life, physical activity, cognitive function. So all of the major secondary outcomes that we're looking at, there was no huge effect on increasing the blood donation. So in some way, reassuring the service actually, even if we want to increase the frequency of blood donation, it's quite relatively safe to do that as well for a shorter period of time, let's say 2 to 4 years. There were as well some increase in terms of symptoms, reported symptoms. So donors tend to report a bit more fatigue, a bit of increased frequency of what is called restless leg syndrome. So some potential symptoms, potentially related to blood donation, but those were relatively minor compared to the potential other effect that we have investigated that actually show there's no major difference whether or not you decrease blood donation. So the next slide, please. So I think the message that we had at the end of the trial was really of keep calm and keep donating blood. Donating blood is an essential part of the blood service. It's key to treat and life and actually to make people sort of living longer in many different diseases, from cancer, but as well from sickle cell, thalassemia. This is actually really relying on blood donation. So what happened after the trial? So the service actually implemented our suggestions. So actually that by keeping the threshold, keeping the interval as the currently is, but actually keeping the option that in case of emergency, there could be the option to decrease the interval, so increase the frequency of blood donation if there's a need of blood for a short period of time. So with saying this, I'll stop here and I'll pass over to Lois.
Okay, thanks, Emanuele. All right, so we've just been hearing about how the interval study has enabled us to see that it's safe and acceptable to bring people back more quickly than the current policy. And we're now going to have a little look to see what might happen if actually we have a personalised strategy for that. So that safety applied to the whole group, but it may be that some people can come back quicker and others need a little longer to wait before they can be brought back. Next slide, please. So this is a schematic of the current blood donation policy in the UK for women. So people come along to their blood donor centre and they have their haemoglobin tested on site. If the haemoglobin level is above 12.5 grams per decilitre for women, then they are able to donate when they come that day, and they will have to wait 16 weeks before they're eligible to return again. However, if your haemoglobin is below 12.5 on that day, you need to be deferred, so you can't donate on that day. Next slide, please. So this is an undesirable outcome, and that's really what we're going to focus on with this personalised recall strategy, is what we can do to minimise those deferrals. Deferrals like this waste the donor's time because they've obviously come along to the centre to donate and then found that they're not eligible to do so on that day. It also wastes the appointment slot from the blood donor service perspective, slot that could have been given to somebody who could donate. And we also know that many donors who are deferred feel demoralised and are less likely to return in the future. Next slide, please. So we're looking to reduce these deferrals whilst also having a minimum effect on what happens in terms of the total donations to the blood service. So one way we can do that is to look at using predicted haemoglobin over time to determine when donors should ideally return on a personal level. So this is a schematic of the potential new strategy. The donor comes along to the centre, and based on the information we already had about them, we know that they should be fine to donate, and we'll come back in a bit more detail to what I mean by that later. Part of that sample is then tested off-site after the donation is made to see what the haemoglobin levels are, and that information is then used to determine when that individual should return next. Next slide, please. So to inform what we're going to do, the work we're going to do on this, we take some data from a study called COMPARE, and we have about 9,000 women in this study. And of those 9,000, about 6,500 returned for a second donation during the 1 year of follow-up that we have. Of those women, 8% were deferred when they attended for low haemoglobin. 3% were deferred for other reasons, so that might have been that they've travelled abroad recently to particular countries, for example. 14% donated, but were later found to be actually less than 12.5 grams per decilitre. So that might be because they were very close to that threshold and there is some measurement error involved in taking that test on site. And 75% donated and were in fact above 12.5 grams per decilitre when they were checked with more accurate measurements off-site later. So we're going to use the information from these 9,000 women to fit some statistical models to look at what happens to haemoglobin recovery after a donation. And these models will account for various personal characteristics, including age and the haemoglobin at their baseline donation. Next slide, please. All right, so this is a figure that shows what might happen to somebody's haemoglobin over time after initial donation. For this particular hypothetical person, their initial haemoglobin is 13 grams per decilitre and they're in the age range 35 to 44. So we can see that they come along and they make a donation and their haemoglobin initially drops dramatically after their donation, and then there's a recovery over time. So this figure is showing what happens under the current strategy. So we're going to recall this individual at 16 weeks, and you can see there the red dot showing their first recall donation. And then they drop again and recover and drop and recover. And as we get to that third recall, close to a year there, this individual now has very low haemoglobin and they're going to be deferred at this third recall. Next slide, please. So what we're looking to do is to optimise when those recall times might be for this particular individual. So the blue line then here shows what those optimal times might look like. So the blue dots showing their recall times. So for this person, actually they could be recalled early in the 16 weeks the first time, but then they need a little longer for their haemoglobin to recover to an acceptable level, and we don't recall them again until nearly 50 weeks. Next slide, please. So one question you might want to ask is why we haven't recalled them at this time point indicated here at about 28 weeks, when we can see their haemoglobin has met the threshold of 12.5 grams per decilitre. And that's because we're using these statistical models to estimate, give our best estimate as to what's happening to this person's haemoglobin, and it's not known with complete certainty. Next slide, please. So what we want to do is wait until we're happy that their probability of being over that threshold is high enough. And for this particular plot, we're showing where we would recall them if we were to accept a probability of 0.8, so 80% probability of being over the threshold of 12.5. And in other words, that they're over that threshold and it's safe for them to donate. Next slide, please. So that probability of being over the crucial threshold to donate increases gradually over time after someone's initial donation. And this shows an example of what that recovery in terms of the probability looks like. So if we were willing to accept an 80% probability that somebody's over that threshold, then we could recall them at 12 weeks in this case for this person. If we wanted that probability to be 90%, so we want to be a lot more certain that they are in fact over that donation threshold, then we would have to wait till 20 weeks for this person. Next slide, please. So how that probability recovers over time, it depends a little on age. So these are all plots showing somebody who comes to donate initially with a hemoglobin level of 13 grams per decilitre. And you can see the different ages there. And for somebody who's in the older age group, they have a higher probability at any given time point of being over that threshold to donate. Next slide, please. However, the probability depends a lot on what your initial hemoglobin was. So for somebody who had only just met the threshold to donate at their initial donation of 12.5, that's the bottom blue line there, it takes a very long time for their probability to be over that threshold to donate again, recovers to a sufficiently high level. But if you were 13.5 at your initial donation, then you would almost always be happy to recall that person, even only after a couple of weeks. However, we perhaps don't want to do that, so we'll come on to that now. Next slide, please. We are going to look at a number of different strategies and what that might mean for the blood service and for the individual donors. So we're going to restrict the minimum recall time to be either 12 weeks or 16 weeks. So not recalling people any earlier than 12 weeks or 16 weeks, depending on the model. And we're also going to set those probabilities of being over that threshold. And these percentages given here correspond to 70%, 80% and 90% chance of being over the threshold to donate. So we'll model all of those things for a population of people and see what the outcomes are in terms of how many deferrals we observe, how many donations are made in donors who in fact had hemoglobin less than 12.5 grams per decilitre, and the total number of pints donated. Next slide, please. So what we do is we model over 1 year what might happen to 1,000 women if we implemented each of those different strategies, new policies. So these results here are based on a 16-week minimum recall, personalised for each person based on those recovery trajectories that we just saw for hemoglobin over time. So you can see there on the right-hand side in the red, these are the results for what would happen under the current policy to compare to. And then we've got each of the different probabilities that we would set for 70%, 80% and 90% probability of being over the donation threshold. So as you would expect, as you require more certainty that somebody has high enough hemoglobin, then you get fewer recalls and also fewer donations over the 1 year follow-up period here compared to what's currently happening. However, we also get fewer undesirable outcomes, by which I mean recall donations that actually were below the threshold to donate. So if we require somebody to be they have a 90% probability that they were over the threshold, then we get obviously fewer people who did donate under the threshold. And we also get no deferrals under any of these strategies because we set that to be the case because we're going to test their hemoglobin off-site. So the strategy that we would prefer here really depends on how we balance those things. How do we balance the total number of donations to the blood service with the deferrals, which is an undesirable outcome, and the recall donations under the threshold, which is also an undesirable outcome. So on the bottom row there, I have given a measure of donations per adverse event, which kind of gives a way of balancing those things. So under the status quo then, we get 3 donations for every adverse event that occurs in these 1,000 women over a year. And this improves over each of those other strategies that we've looked at up until nearly 15 donations per adverse event when we require the probability to be 90%. Next slide, please. And these are the results where we have the last ones were 12 weeks. These ones are 16 weeks. I think I had written 16 on both. So for these 12-week results, then we get obviously quite similar, so because not many people can be recalled as early as 12 weeks. If we can just flick back to the previous slide. So if we compare the 90% strategy there, we get 14.8 donations per adverse event. And then if we reduce the minimum recall to 12 weeks, next slide, compare that 14.8 to here, 14.2. So actually, if we allow people to recall as early as 12 weeks, we end up over a period of a year, actually with not as many donations per adverse event. And that will be perhaps because some people are taking a lot longer to recover from that early, quick recall donation. Okay, next slide, please. All right, so I have a few assumptions that have gone, obviously a lot of assumptions that have gone into this work. Mostly though, these things are relating to the data that we have used to inform the statistical models. One of the key points is that people returned in the informing data, in the data from COMPARE, over a range of time points. And we have assumed here that that return time that those individuals chose was not related to their hemoglobin level. We also use data from just one return visit. We only have one return visit in COMPARE, not multiple return visits. And we apply it here to allow people to come back multiple times over 1 year. Additionally, we assume that there is a linear hemoglobin recovery trajectory for the recall times that we are interested in. So it's very possible that the recovery trajectory is not linear in the early part, but we are not recalling people as early as 2, 3, 4, up to 12 weeks. So it's very likely that the assumption of linearity is not unreasonable in that later part of the recovery that we're looking at. I think the overall message here is that a personalised strategy may allow some people to be recalled safely early, whereas other people may need to wait longer. But overall, this strategy is likely to offer the blood service a good compromise between the amount of blood collected whilst also reducing the number of deferrals. Okay, thank you. I'm going to pass over to Nick.
Hello, everyone. My name's Nick. I'm a researcher here at the University of Cambridge. Firstly, thanks all for coming on a lovely Tuesday evening to listen to our talks. And so what I'm going to talk to you about this evening is something called donor genotyping, which is actually typing blood groups of donors using their DNA. So next slide, please. So I'm going to assume that you don't know so much about transfusion medicine and also about donor genotyping. And I'll start with the very basics. So there's a number of reasons people might be in need of blood or might be losing blood. So there's hemorrhage, they've been in an accident or during childbirth, you can lose blood, or abnormal hemolysis due to cancer and infection, and various genetic diseases, so inherited human disorders like thalassemia and sickle cell disease. And basically, these always kind of follow a similar pattern where there's disruption of the oxygen transport system in the body, and it can result in organ failure and death. And there's also, from destruction of red cells, release of iron, which is inside your red cells, which can be highly toxic. So what can we do when people are losing blood and in need of blood? So next slide, please, Sarah. So a blood transfusion, I'm sure you've all heard of blood transfusion, but it's basically the transfer of red blood cells or erythrocytes from one individual to another. And it's a widely used and effective clinical intervention. And this is actually, I think, a transfusion the patient is laying down by the looks of it. Next slide, please, Sarah. So transfusion, although it's very, very widely used in many areas of medicine, it's not without complication. So next slide. And one of the main complications is called Aloe immunization. And what this basically is that there's antigens present on the surface of your red blood cells, which the immune system can recognize. And if you're exposed via a transfusion like havent or during pregnancy to blood with antigens that you don't possess, then that can trigger an immune response because it's your immune system seeing something that it doesn't recognize. Next slide. And just to highlight how serious Aloe immunizations are, they can cause hemolytic transfusion reactions. So this tube here that I've got on the slide, that's roughly 10 mils of blood, and just 10 mils of blood mismatched for ABO blood group, which I'm sure you've all heard of, is enough to kill you. And if you have a mismatch for another blood group system that isn't going to cause a very severe reaction, the antibodies produced by your immune system from the mismatch will persist, and they can cause complications in further blood transfusions where you have a much stronger response. So next slide. And so really this leads to kind of like this golden rule in transfusion medicine that in order to ensure the safety of a blood transfusion, it's really critical to identify the clinically important blood groups of both donor and recipient. So to match the blood between donor and recipient. Next. And so how do we identify blood groups and how do we match people? Well, for the longest time, we've been using something called serological typing or antibody-based typing. And there's a lot of complex methods like the Coombs test and indirect antiglobulin test and automated methods, but they kind of boil down to this, which is that you take some red cells, which I've represented with the big red circles in this cartoon, and you take an antibody that's specific to something. So it's specific to blue. The blue dots are what the antibody is specific to. And you mix the antibody in the red cells, and you can see that on the top row, the negative red cells that don't have any of the blue antigen on their surface, they don't agglutinate. So the tube, if you imagine, stays milky red. But if they are positive for the antigen you're typing for, then they'll clump together and sink as a pellet to the bottom. And for the longest time, this is how we've been typing blood donors. Next slide. And it's seen as a bit of the gold standard, but it does have some limitations. And so what I've shown you here, so this table that I've shown you here, it's just a selection of each column represents a different blood group antigen. So something that you might want to type for. And I've kind of selected the ones that I would say were commonly typed. And what the color represents in each box is the presence or absence of a typing result. So not whether you're positive or negative for that blood group, but just whether somebody's been tested. And you can see here, the data is very patchy. And it turns out that only 85% of donors, so 85% of donors don't have any typing for any of these commonly typed for antigens, and about 95% of donors don't have any typing for the very, very rare blood group antigen. So we just don't know if they've got one of these rare blood groups. And this is for a couple of reasons. But the main ones is that for some of the blood group systems, there is no high throughput method. So there's no cost effective or quick way of typing thousands of blood group donors. And also another big limitation of serology is that after someone's received blood, if they've had a mismatch, when you've got a mixed blood sample, it's a nightmare to identify what is the patient's blood group and donor's blood group and what's going on in that mismatch. Next slide. So what can we do about this? Well, it turns out that the genetics of blood groups has been studied for like the past 50 years in great detail, and we know a lot about it. And so what I've tried to show here is like the little, this is called a Circos plot. Around the edge, each one of these labels represents a different gene. And they actually underpin a blood group, one of the blood groups, and it's actually outdated. So I think there's 38 on here, but there's actually 40 blood groups recognized now. And the blue is actually platelet antigens, which is a different type of antigen system that's not relevant to blood groups specifically. But really what I want to get across is that we know the link between the DNA code and the blood group of a person. So it should theoretically be possible to identify a person's blood groups using their DNA rather than using antibodies. So next slide. And basically from this idea and with genetic technologies, so genetic DNA typing technologies becoming much, much cheaper, The Blood Transfusion Genomics Consortium, which links several countries from all around the world, was set up in order to try and develop a DNA-based test that could type human blood groups to clinical standards. And what I'm going to talk about for the rest of the talk is actually the work that we've been doing in the BGC and where we're kind of at now. So next slide. So essentially what this whole project boils down to, if you look over on the left, you have DNA type information from one of these tests. And we run that DNA typing through an analysis algorithm and a database that really collects the kind of cumulative knowledge of blood group genetics and blood type genetics, and sort of combines them and then prints off a blood report that can be used, you know, in a blood bank or to type donors or patients. Next slide. And when I talk about genetic tests, I thought I'd better introduce the actual test itself. So what I've been really focusing on is the UK, something called the UK Biobank array, or Axiom array is the official name. And what this test does, so you can see this little plate here. So that's 96 little glass slides. And on the top of the glass slides are little probes that measure a specific position, a specific DNA base, so A, T, C, or G, in your genome. And we measure 800,000 of those positions on each one of these tests. And the tests are actually highly customizable, so you can change around which positions you're measuring. And the beauty of these tests is that they've already been used in large-scale studies that the previous two talkers were talking about, to type around, you know, just over 500,000 individuals. And a lot of those are blood donors. So it really gave me access to a set of genetic typing results or DNA typing results and blood typing results to go ahead with this study. So next slide, please. And so what's on these arrays? What are these 800,000 positions? Well, we've been working to kind of customize them specifically for blood donor typing and blood donor relevant things. So you can see that I've drawn a cartoon of the array here, and you can imagine that the magnifying glass is showing you the part that types blood groups, but there's also lots of other stuff on there. So there's, you know, content, or probe content, we call it, for tissue matching and transplantation, iron accumulation disorders, red cell quality, all sorts of things that are very relevant in transfusion and transplantation. And so this is kind of the technology that we want to move forwards and say, okay, these are very, very cheap tests that can be used to screen thousands of people when there's thousands of blood donors required every year. How well do they perform at this? So next slide. And so our project was basically followed this kind of schematic diagram, but the aim, like I said, was to develop a comprehensive platform for donor typing and genotyping that performs to clinical standard. And what we've done over the past few years is we've taken a set of donors, typed them on a version of the array that we've designed. It used the algorithm to impute their blood groups from the data, and then compared that back to their current typing that's already on donor record in NHS Blood and Transplant. And then we've seen where we've made errors, we've understood the errors, redesigned, and then done another validation on a new set of blood donors. So next slide, please. Sorry. And so we've really started, and so last year we published a paper in which we did the final sort of evaluation and proof of principle, and we used DNA from just under 7,500 blood donors from the UK and from the Netherlands. And what I'm showing you here in these heat maps is the typing data that we get back. So each row in one of these heat maps represents a different blood type antigen, so something that we want to type for. And if you look at the top two squares, they represent what's currently available in the blood service. So remember, I showed you in one of my first slides this Swiss cheese kind of holy patchy data. So that's what it looks like en masse when you look at a large number of donors. And what is on the bottom two squares is the blood typing results you get back when you use a genotyping test like the ones I've been describing. And what you can see is that there's just really confluent and complete data on everybody. There's a few places missing where the test probably didn't type as accurately, so we've excluded a result. But overall, we got a 3.8-fold increase in the number of typing results available. So it went from 12 typing results on average available per person to 47. And that's only for the antigens that we were able to compare. So we were only able to compare 48 antigens because that's all there was on previous donor record. We actually have further data in the genetic type or DNA-based typing results for 198 additional antigens that are clinically, some of them are clinically important. So next slide. So you might say, great, Nick, you can make hundreds of thousands of typing results, but are they actually accurate? And obviously this is a question that we set out to address. And so in the clinical typing, there was 58 antigens available and 96,000 results. And in the array-based typing, the DNA-based typing, there were 224 antigens and 1.6 million results. And when you overlap those, you are able to perform a comparison, a high level of comparisons for each antigen for 38 blood type antigens. And this means that we compared 92,000 results, and we only had 72 errors in those 92,000 comparisons. And this plot here, basically, so on the y-axis, on the vertical axis, you can see it's percentage of the total comparisons for each antigen, and on the x-axis we have each antigen. And you can see it starts at 90% to 100%. So this is a sort of zoomed in plot on the very top. And you can see there's a very low error rate spread across some of these antigens. And what I haven't showed here is that obviously we investigated these 72 discordances. And again, like I said, we understand most of them. And it actually really boiled down to 50% were errors that the DNA typing test made, and 50% roughly were errors that were made in the serological typing tests. And so they really complement each other actually on the types of error that they said there. So far I've shown you we can make hundreds of thousands of typing results of those tests. They perform equivalently with current methods regarding accuracy. And so next slide. What are the benefits? You say you have all these accurate typing results, but what actually are they actually any use? You know. And so one of the ways that we tried to prove that was we took NHSBT complex case referral data. And what this is people who've had mismatches, multiple mismatches, and generated 2 or more Aloe antibodies. And that means that they have to have special matching requirements because they have antibodies against multiple blood groups. So you have to take really great care when you're giving them their next transfusion. And we used the 7,500 donors in this study as a sort of virtual stock. And we said, okay, how many times can we match them using the currently available data? And then how many times can we provide a match using the same donors, but using DNA typing data on top of the currently available data? And we actually found that there was a 2.8-fold increase in the number of kind of matches we were able to identify in the same people. So they were much more useful. And what I've showed here on the plot is that on the y-axis, vertical axis, is the average increase in available donors. And across the bottom is the Aloe antibody count. So almost a rough marker of complexity of the transfusion case. And so you can see there's this big increase when there's 3 Aloe antibodies and tailing off, getting more and more difficult to match as we approach 9 Aloe antibodies, which is a lot of Aloe antibodies, actually. It's very rare for someone to have that many. And it represented in this set 176 additional patients, new patients who now could have a match of blood using the same donors. So you've really increased the value of those donors. Next slide, please. Thanks. And then this is sort of a zoomed in plot of the last plot. So across the bottom is the number of times a different combination of specific Aloe antibodies or specific matching requirements has been seen. And on the y-axis again is the increase in available donors. And what I've tried to do here is like zoom in and show you some of them. So if we look at the very right hand side, you see this A negative and anti-C, anti-D, anti-E. So that's one set of requirements. And you can see that genotyping really, for the combinations where we know those, we see those antibodies a lot, we're performing about equivalently. And that's because that's kind of what we're set up to do at the moment. But if you look over on the right, the person with 9 Aloe antibodies and also the person way up at the top there the A-pos and anti-COB, anti-K, anti-WRA, you can see there's thousands more donors available, and that's because we don't routinely type for anti-WRA. And this is the kind of advantage of these cheap DNA screening tests, that you're now able to make much better use of your donors. So next slide, please. And like I said, oh, one more previous, if that's all right. Yeah, so this little caption at the bottom, basically what we can kind of imagine is that we'll move to a stage where lots of people will be typed very, very extensively. And instead of, you know, matching as best we can, and then there's some immunization happening just in the system, actually we can preemptively, much more extendedly match people. And it means that we might reach a point where we no longer have to accept allo immunization as an unavoidable consequence of transfusions. We might be able to, you know, sort of counter that by extended matching people, and there's a lot of work going into that at the moment. So next slide, please. And so I just wanted to get across to you, maybe people who aren't in the genomics community, the future scale of this analysis. So this news clipping is getting quite old now. It's 2018, but the UK did commit to sequencing 5 million genomes within the next 5 years, and they're well underway with that. And so, you know, this blood typing information can be extracted from this genetic data. And next slide. And then there's also other huge studies going on where, so the last one was mainly focusing on patients, but the UK Biobank is a resource typing, you know, healthy individuals and looking at the genetic link between genetics and then normal biological traits. And that, you know, in 2019, that project announced it was going to do another 500,000 genomes. So I really wanted to get across that this data is becoming more and more common, and that we should be extracting as much value out of it as possible. And that means, you know, typing blood donors who've joined these studies and bringing the results back in so that it can be confirmed within the blood service. Next slide. And I just wanted to show, this is a very quick slide. It's just to show that, there was a pilot study in which 10,000 individuals were sequenced by the NHS, and we ran it through our algorithms. You know, we were able to identify 737 O negative people, which is an in-demand blood type. But more importantly, is the sort of middle region. We were able to identify 140 individuals for 18 different antigens, which are extremely rare blood groups. Like U negative is rare, it's in high demand. RH26 is in high demand, and things like this. And so we had estimates, you know, obviously went around some of the blood banks, says how much would it cost you to type this many people? And it was, you know, many, many thousands using current methods, and many, many man out that, you know, very labor intensive, lots of hours going into that. Whereas this test can be done in an automated, very high throughput fashion. And it's a brilliant screening tool. Next slide. And really, once we started doing blood groups, we started saying, okay, well, what other antigens can we type? And we looked at stem cell and organ donation typing. So it's called HLA typing is how we refer to it. And it's the same DNA test. So you're not paying for a different test. It's the same test. And it's the same donors. And what we'd managed to do is we managed to find a subset of a subset of a few thousand individuals who also had some HLA typing available. And we did 9,000 comparisons to HLA typing. And as you can see here, we got 99.3% concordance at a resolution, which means these people can now be used for, you know, supporting cancer patients who need stem cell transplants. And next slide. And so that's highly relevant. That we can do HLA typing. So I don't know if you remember this story, but there was a young child called Oscar who really needed a stem cell transplant. You know, there were people queuing to come and donate because he had a very, very rare HLA type, I think. And the message I'm trying to get across is that as the more people join these studies, and if we start integrating these tests into routine donor typing, you know, there might be a situation where we can find these rare types in a much more cost-effective way and much easier to provide care for people. Next slide. So further away, so we're at the point now where we have a comprehensive platform for universal blood donor typing. And so really the next steps are to kind of get NHSBT, and I say Sanquin, which is the Dutch blood service here, set up for collecting this data from these national resources and national projects, and set up to do their own typing. And obviously we want to do a clinical trial of better matched blood, so using this genotyping or DNA based data to provide a better match for people. I don't think, is that a next slide? I think this might be the end of my talk. Yeah, so that's the end of my talk. And I just want to say thank you to you all for listening. And then also to Sarah for managing all this and bringing it together for us and doing the slides. So brilliant, thanks.
Excellent, thanks Nick, and thanks Lois. I think just before going to some question, really thanking blood donors that have been really sort of helpful with us and donating their blood and their information to perform all our research, and as well NHSBT and NIHR that actually have been funding a lot of our research, and as well many of our colleagues actually help to produce and to analyze and to make this research available, particularly in the BTRU in Cambridge, but as well in other part of University of Cambridge, Oxford, Sanger and Sanquin. So I'll stop here, and then we have time for some questions. So I think the way, if you want to either raise your hand or put your question in the chat, then I can call you on you or can read the question.
I think there's a question in the chat, Emanuele, which is,
yeah.
Why is there such a low amount of blood donations in the London area, which is the map in the first part of the presentation?
So I think so. There actually, there was not a low number of blood donors. I think the number of blood donors is actually the same in different parts of the country. I think the number of blood centers actually recruited in interval study, there are not many in London. So in London, there's many, so interval recruited donors through what is called the fixed center, where people can go and donate, while there are many more mobile teams that actually go around London in different locations where blood is collected. So that was a reflection, I guess, of the element of the study, not of the blood donation itself. So the second question, I guess, is around iron status and ferritin, and how hemoglobin level is actually not a good predictor of iron store. I think that's it's a good question. It's a good point. The blood service usually use hemoglobin level to screen donors to detect iron deficiency, basically. And this is usually done because it's very cheap and quick method that can be done. It can be scaled up to many millions actually of donation and donors. And this is done the same way through many of the services around the world. I think what some services actually implementing is supplementing this measurement with a measure of ferritin that actually provide information about your iron store. One of the issues with measuring ferritin is that ferritin is not available to measure on the spot. So there's no point of care testing to measure ferritin, and so to allow that, you can't allow donors then to donate when they're coming and donating the blood service. So I think it's there's a bit of a difference between measuring hemoglobin or iron store, but I think hemoglobin is a good, in some ways, not perfect, but it's a good predictor of what will your iron store will be, and so it's used as a proxy for that.
Maybe if can I take that question, the last question?
Sure, yeah.
So the question is, what are the key challenges to overcome to type blood donors routinely? So if I wasn't clear in my talk, they are typed routinely using the serological methods, but the biggest challenge to implementing the genetic typing methods is that the test, first of all, needs some sort of accreditation from an official body. So we did a proof of principle, but to make it so that blood services can really easily adopt it and produce clinically, clinical grade, high grade, very accurate results, it needs accreditation. And so the BGC is currently running an accreditation study where we're collecting a sample panel of 15,000 DNA samples from all over the world, all the members, and they're going to submit that, and we're going to genotype it in triplicate, and then we're going to seek accreditation from the appropriate authorities and help the blood services to bring the technology inside their into their organization so that this can be done in a routine way.
And I'll take possibly the next question is really about why do we donate all the same amount, and can we tailor the amount of blood donated to different sides of each of us? And that's another very good point. I think there are some countries, especially in East Asia, in which the donation is slightly smaller, given that usually the size of men and women is slightly smaller in those countries. But in general, I think this is not done, because one of the issues then the amount of blood that could be transfused and the risk of exposing donor recipient to more than one bag of blood if they just need one bag. So exposing more people to more donors compared to fewer number of donors. So I think this is something that hasn't yet been sort of evaluated properly, I will say, but some countries already are taking this approach, and I guess this is something that would be good to study possibly in future research. Any additional question from the audience?
So a lady who is A negative. So A negative represents two blood groups. So A is from the ABO blood group, and the negative is a blood group. It used to be called rhesus, but we call it RH now. So it's RH negative or D negative. So that minus represents D positive or negative. And ladies who are pregnant with a baby that is D positive, and they are D negative, some of the blood during the first childbirth, what happens is some D antibodies, some D positive blood cells can be exposed. The mum can be exposed to some D positive blood cells. And what this means is that she can stimulate an immune response, and then in the next pregnancy, if the baby is positive, some of the mum's antibodies can, cause damage to the baby. So to prevent that initial immune reaction, you give anti-D injection, which kind of like soaks up all the first baby's blood cells, so that there's less of a chance of the immune system generating antibodies and recognizing in future pregnancies. That wasn't the clearest explanation, but that's the basic principles.
Thanks, Nick. Any additional one? If not, well, thanks again for joining us. I hope you enjoy our seminar. I think there's some question you have to answer before exiting the Zoom room. And I hope to see you all next year. I hope to see you all in person next year for another series of our seminar around blood donation research.